Hi, everybody. Welcome to our today's lecture. So we are going to focus on cell division today for what we consider our somatic cells, which are basically any cells we have in our body, minus what we consider our gametes or sex cells, which is the sperm and egg. Some of the learning outcome, again, looking at the process of cell division, looking at how these processes take place in sense of chromosomes or genetic information being divided. What mechanism do we use to control the process of cell cycle? And basically what happens if there's problem with the actual cell. We undergo a process called apoptosis. So those would be some of the main focuses of our class lecture today. So when we talk about cell division, the basic idea is we are looking at taking one original cell and making copies of that cell. Now, we do have two processes that cell division uses. One is mitosis, used by our somatic cells, and that is creating two identical daughter cells at the end of the process. Versus when we're talking about meiosis, we're actually looking at creating four cells that are not identical to one another, which is going to be eventually used during the process of gamete development and basically creating the offspring from the parents. Now, again, this lecture is focusing on cell division related to mitosis of the somatic cells. So just kind of giving you some information, our bodies consist of about 30 trillion cells, which is a huge number if you kind of imagine the numbers, billions and then trillions. And we continuously create new cells, for most part, to replace the old cells that have died out. And it is important to note that there are cells in our body that basically undergo cell division at different rates. There are cells that you're basically born with and die with. There is no regenerative power in those cells, so they do not undergo cell divisions. And then there are ones that continuously undergoing cell divisions, like the cells that line your stomach, or your skin cells, or your hair and nail cells. So these are continuously growing and undergoing cell division. Another purpose of having cell division is basically growth. So if you think about you starting as a single cell organism during the process of fertilization, that single cell undergo regular cell division, making one cell to 2, 4, 8, 16, 32, and so on and so forth, until you end up basically with millions or 10 billions and then eventually trillions of cells. And again, that's during the fetal development, formation of basically a baby being born and undergoing the stages of development all the way to adulthood. Maintaining health, basically, again, a lot of these cells are ensuring that you are staying healthy. For instance, the lining of the stomach needs to be continuously regenerated. Otherwise, the acidic environment of the stomach causes conditions like ulcers. If your skins are not regenerated properly, skin cells are not regenerated properly, then you end up with very skin cells and very susceptible to skin damage, cuts, and basically breaking that boundary to the external environment. Healing from disease and injury also requires this process of cell division. Again, kind of thinking about if you cut yourself, the fact that you produce a scab and then eventually that scab falls out and you have beautiful skin underneath it, which is brand new, that's also a process of cell division. Now, when we're looking at cell division, the initial process is a process called interphase, which I'm going to talk about in detail. And in case of our somatic cells, again, every cell in our body, with the exception of the gamete, interphase goes to mitosis, and then mitosis goes to our cytokinesis, which is, simply put, you will start with one cell, by the end of this process, you have two identical daughter cells. Now, again, as I mentioned earlier, we do have a process called cell death. This is processes where the cell basically get double checked for the accuracy of the genetic information. And in most cases, if there is any major changes or difference or mutation has happened, then those get tagged for destruction through the process of apoptosis. And we'll talk about it at the end of the lecture. Now, when we're talking about mitosis and apoptosis in terms of our regular cell activity, it is important to understand that your cells numbers are going to go up during the process of mitosis, as depicted here. You start with 1 cell and you end up in this picture with 8 cells. And then the opposite process is basically basically breaking down your cells and starting with a large number, and then as time passes by, you destroy some of these cells. Now, again, under normal conditions, we do not want to have a significant apoptosis, or in another sense, apoptosis and mitosis should be relative to one another. For instance, in this case, well, I have shown an image that you can see right here. This is where apoptosis is not happening at its normal rate. So in an embryo, when the fetus is basically, embryo is developing, we have all of our fingers connected together, kind of web-like structures. And as time passes by, basically your cells undergo the apoptosis, allowing these fingers to be individual structures. So you basically think about it as eating the cells that holds the fingers together. So a medical condition called syndactyly is basically when this apoptosis process in the embryonic development has failed, and webbing may persist among some fingers or all of the fingers in the hands. Now, again, as I mentioned earlier, we have the process called cell cycle. So the cycle that you see right here on your image defines the process of cell cycle. So one of the things that you notice if you are looking at the labeling, about 75% of the time of the cell cycle is spent on the process called interphase. So interphase is a very long process, and itself is made up of 3 phases, what we call G1, S phase, and G2. So what are these phases? So during G1 and G2, which is first and the last step of interphase, or subphases, I should say, is where you basically start to grow your cells and make copies of all the structures and organelles that are found in one cell. Again, understand the process of cell cycle is to start with one cell and end up with 2 identical daughter cells. So it is important to have all of the components that are inside the cell already duplicated and ready to be separated or divided between the 2 new cells that are being created. During the S phase, is the way I think about it, S phase stands for synthesis. So this is the phase that your DNA is undergoing replication, where basically you start with your original DNA sequence and you make 2 copies of that genetic information by creating semi-conservative DNA sequences, something we described previously. Again, the goal of having the S phase is that by the end of the process, when you have the 2 cells, the 2 cells will have identical genetic information to the parent cell. Now, it is important to note that not all the cells undergo the interphase and then complete the process of mitosis all the time. Many times, the cell completes its G1 phase and then kind of go into what we call a G0 phase. So G0 phase is basically you have done some monitoring. You either, if there's damage to the cell, you don't need the cell goes ahead and gets destroyed. uh, you can have a cell that remain at this level. It doesn't go any farther, so it's basically stuck in this level. Or you have cells that basically eventually go back into the S phase and continue with the process of cell cycle. So think about it as a timeout phase of the cell cycle. Now, mitosis or mitotic phases are what we describe here in blue. And again, all of them together take about 25% of the cell cycle timing. So in another sense, if you're looking at them, they do happen much more rapidly compared to the interphase, where you're basically utilizing a lot of resources to double the material inside the cell, the structures inside the cell, as well as your DNA. Now, the phases of cell cycle for the mitotic phase are prophase, metaphase, anaphase, and telophase. And again, if you notice, they are about the same amount of time where your cell spent in each of these phases. Also note that you do also have a process called cytokinesis, which is a stage where your cells are fully separated from each other by cutting or separating their plasma membrane. It does not consider part of the mitotic phase, but it is part of the, it is part of the cell cycle. Now, as we go through this a little bit more detail, we're going to take a look at each of these phases, including the interphase and the mitotic phases, and specifically understand how the cell is changing as it passes through each phase. So we're going to start with the interphase, which is going to be the first phase. Now, as we mentioned earlier, interphase has 3 separate subphases, which is G1, S, and G2. This phase is very important because this is where your DNA is being replicated. And again, the whole idea is because I want to go from 1 cell to 2 cells in this phase, I need to double everything that I have inside my cell, including the structures. And as I mentioned earlier, we do have G2, which is kind of thinking about it as a timeout phase where your cell is trying to figure out what to do. Now, as we go through these stages of the development for your cell, one of the phases that pops out is the process, it's development of what we call centrioles. And centrioles are, and I had a picture here, but apparently it's not loading, is these kind of, think about them as these little, I guess, they look like a star if you look at them as a cross section, but if you look at them at the side, they kind of look like these little cube-shaped structures. So if this is my cell, this is kind of my nucleus with genetic information in the form of chromatin, basically doubled during the process of interphase. So centrioles are made up of microtubules. So microtubules, if you guys recall, is one of the 3 cytoskeletal protein structures that we had inside the cell, or cytoskeletons we have the cells. And this is the largest of the 3. And these microtubules, these centrioles in another sense, are eventually going to be utilizing proteins to develop structures called spindle fibers. And the way I usually draw spindle fibers are as these little kind of string things that are coming out. So these would be your spindles. And as we see during the metaphase forward, these spindles are extremely important for movement of the chromosomes in the right area. So you have equal division of genetic information between the 2 created cells. Oh, sorry, here's my picture. I guess I had it on the thing. So here's basically your centrioles that I draw. You see the spindle fibers being developed, and then where I draw on would be your nucleus with the genetic information. Another thing I want to kind of talk about is something we described in detail previously when we looked at the DNA structure, but this is a good chance to review that content. So when your genetic information is present during the process of interphase or the phase interphase, is considered to be very uncondensed because you really at that point don't want to do anything with it. So before, I should say before interphase, if your cell is not undergoing cell cycle, your genetic information is relatively uncondensed. However, as we undergo the process and finishing up the interphase and getting ready to go to the mitotic phase of the cell cycle, that genetic information needs to be condensed. And that's exactly what's going to take place. So one of the things that I want you guys to note is the difference between these two pictures. So if you look at the top picture, you have a chromosome which only has one arm. And this is basically where the genetic information would have not replicated yet. So as we already learned and talked about multiple times, during the S phase of the interphase, the genetic information is being replicated. So when we actually draw a chromosome as that kind of an X shape that we keep drawing, you're actually drawing this structure and this structure. One is the original, and the other one would be the copy. So I want to make sure this is understood that the arms of the chromosomes that we draw as an X is a depiction of the genetic information has already been replicated. This does not happen in interphase. It actually happens in prophase. So again, the goal for you to understand how you go from this kind of uncondensed genetic information to a very condensed genetic information that's observed during the mitotic phases. Another thing that I do want to point out is a couple of terminology. One is that each of the arms of the chromosomes are called sister chromatids. Again, something we discussed before. And the area at the center where the two chromosome arms are basically joining each other, that's referred to as the centromere. So if you're looking at the genetic information in a typical human cell, you will have 46 chromosomes, right? So in an uncondensed version, not replicated, we will have, condensed version, but not replicated, you will have 46 chromosomes. Again, 23 is from your dad and 23 is from your mom, or maternal and paternal. Now, as we undergo the process of DNA replication and condensation of that genetic information, you end up with 92 chromosomes, which is, again, it's referring to the fact that the genetic information has been duplicated. So you have 92 of these arms of the chromosome, which eventually divides to two cells and create a 46 chromosome cell each. And I want to just kind of do a quick depiction of what that looks like. So if you have the original cell with 56 chromosomes, as it's going through the process of interphase and mitosis, I should say just interphase, you're going to end up with 92 chromosomes because that's where genetic information duplicated. And then by the end of mitosis, you're going to have 46 on one cell and again 46. And these two cells are identical in terms of genetic information. Now, prophase. So what do we need to know about prophase? So prophase is very important for us because this is where we're actually transitioning from a uncondensed genetic information, which is chromatin, to a condensed version of the genetic information, which is the chromosome. This is extremely important because if genetic information is not condensed, it's not super tightly wrapped, it can be broken or damaged during the process of cell division. So this is one of the first steps that takes place before we even go further in terms of alignment and pulling things apart. Another thing that we see is development of our spindles. So again, these lines that you show right here, these are our spindles that are being developed from our centrioles. And then another factor that you do want to note is that if you remember your cell, if this was my cell, this was my nucleus, and around the nucleus, I had my nuclear envelope, right? So as the process of prophase takes place, you do want to get access to the genetic information. You do want to divide it between the two cells. So that nuclear envelope basically is in our way. So you do start to degrade it. So you notice that there's big gaps, and eventually this wall, these structures will completely disappear, and basically releasing the chromosome within the cell. Now notice again, as the membrane gets broken down, you don't have any nucleus, so there is no clear nucleolus or nucleus will be visible for us. I don't recall if I mentioned this, but nucleolus is the one that is involved in our ribosome synthesis. So if you guys don't recall it, or if I didn't mention it, here is the information. So nucleolus is a structure inside the nucleus, which is this one. I'm going to show it to you, visualizing it. Essential for ribosomal ribosome synthesis, which is the small and large subunit. But as I mentioned, we don't need to do any protein synthesis at this phase. We just want to divide genetic information. So the next thing that happens in the mitosis phase is metaphase. So metaphase is a relatively easy process. What happens is that the spindles that you have come in and connects to the centromeres. So remember, those centromeres are that point of attachment for the two sister chromatids. And as these spindles elongate and as they move the nucleus, the chromosomes with them. And what they do is they actually line up the genetic information right at the center of the cell. And it is also important because I think it's interesting if you were to look at a cell, if you're looking at a cell undergoing mitosis, during metaphase, it appears that the chromosomes are just staying there. So they appear actually motionless, but in reality, you have a lot of tension that comes from the spindles, basically making sure that alignment is taking place. So they are being pulled, but the pulling and the force is equal on either side, therefore ensuring that the chromosomes stay in the middle or the equator of the cell. By the way, if you're wondering what you're seeing in terms of this beautiful coloring, if it's not clear, what do we have in blue? Blue is representation of the chromosomes. The green is representation of the spindle. And basically, this is the cell itself. So basically, any structures within the cell. Now, as of right now, we haven't separated any genetic information. So that's what takes place in the anaphase. So what happens in anaphase is that the centromeres start to divide. So the centromeres was the point of attachment, right? So if this was my X, this was the centromere. Centromeres are slowly start to fall apart. And that allows the tension that's being placed on the chromosome to be released. Now, as the chromosome separates, now you're going to end up with two separate arms of the chromosome. And they, at this point, now they're going to be moving into the opposite side or opposite directions of the cell. So what takes place here is this is a very kind of a brief phase. And if you notice right here, which I think is really cool, these spindles are pulling half of that chromosome to one side and half of that chromosome, the X chromosome, to the other half. And based on the coloring, you also notice that they are the same shape and the same color, which means the information on either cell is going to be identical. Another thing that I do want you guys to note is that anaphase is where the cytokinesis starts to take place. It's initiated, it doesn't finish until all the way after the telophase has been done. But it does initiate in the anaphase. Now, the other thing that we want to kind of, the last step of this mitotic phase is the telophase. So telophase is actually important for us because at this point, your cell has its genetic information divided, and it's ready to basically finish up the process. So at this point, I do not need chromosomes anymore because the force that was needed to pull them apart, which put the DNA in danger of being damaged, has already passed. So I don't need to keep the chromosome as their condensed version. So one of the first thing that happens is that the genetic information starts to return back to chromatin. So you start to basically relax or uncondense the genetic information. Another thing that happens is the job of the spindles is also done. So I don't need those spindles anymore. I don't need those spindles anymore. So I can get rid of them. So you notice that they're kind of reverting back and they're getting broken down. Now, as the genetic information goes from chromosome to chromatin, remember, chromatin is fragile, which means that I need to start to protect it. So I'm going to start to reform the nuclear envelope. As you see, that it starts to reappear. And one of the last things that happens is the nucleolus for each of the cell starts to reform. So that cell basically gains its ability to form ribosomal subunits and eventually make proteins for the cell. Now, at this point, you are done. Genetic information has been divided between the two cells. And now all I need to do to finish the process is literally separate the plasma membrane and create two identical daughter cells. So if you guys note here, you do have these invaginations happening right here, which is part of the process of cell cycle. This is referred to as the cleavage. So cytokinesis is basically what happens. Sorry, my computer's acting up. My cytokinesis is I separate that plasma membrane that was holding those two cells together. And that's marks the ending of the cell cycle. Notice that I mentioned that organelles and macromolecules are distributed equally between the two daughter cells. Now, it's not going to be perfect, but it's going to be very close between the two cells. And then all the microfilament bands finally contract and basically finish and pull the two cells apart, making two identical daughter cells. So here in this picture, you can see your nucleus as a very visible structure, which is, again, completely almost separated. So this is the last point of attachment for them. So if I ask you guys the question of what are the steps in a cell cycle related to somatic cells, we start with interphase, and then you have the mitotic phase, which has prophase, metaphase, anaphase, and telophase. Please make sure you can make that distinction between the interphase and the mitotic phase and these divisions. Now, as we understand, what we understand about the cell is your cell doesn't just go ahead and say, okay, I've divided my genetic information, I'm set to go, let's divide the cells. They are a lot of checkpoints and checking and double checking, I think about it, to make sure that everything is okay with the cell before we move on. So basically, as I said, control of mitosis is a daunting task. If you too little and an injury goes unrepaired. So if you don't allow the cell cycle to happen, if you don't allow the cell cycle to continue processing, you may actually end up with tissues that are not being repaired because the cell cycle doesn't happen as fast as it needs to be. However, what happens if I have cell cycle happening too frequently, too many times? That causes abnormal growth, which is, again, not a good thing. What this means, I do not necessarily mean cancer. I just simply mean that there is an abnormal growth that could be just simply a tumor that is not, does not consider cancerous. Now, there are groups of interacting proteins that are activated during specific times within the cell cycle. And these interacting proteins are basically going to be the proteins that carry out the process of our checkpoints. In another sense, they're making sure that what we have accomplished so far is correct and there is no problem before we move on to the next step. One of the most important things that we want to again focus on is making sure that the chromosomal information has been replicated correctly and then divided equally between the two cells. They are both internal and external components and factors that can affect the mitotic clock. Again, how fast or how slow that cell cycle takes place. Here is an idea of what those checkpoint looks like. We already talked about a couple of them. So for instance, if you go here, if you're at G1, you can actually go to the G0 phase, which is stops the process, unless your cell wants to go through the cell division for whatever reason. During the S phase, which is basically where you are replicating DNA, we really want to be careful, right? Because if the DNA is damaged, that means the cell is not going to work properly. We're going to slow down the process, allowing for checking of the DNA. During G2 is also another stop, is basically ensuring that the genetic information has been double-checked before we move further. Another checkpoint happens right before you finish the interphase and start your mitotic phase. If there is, again, any abnormality in the genetic information noted, the cell is forced into apoptosis, which means basically cell death. Now, if you have a proper genetic information, everything checked, and again, double checked, then you introduce your cell to the process of prophase. As the spindles are starting to form and pull the genetic information in the right direction, we do check the spindle assembly to make sure that the DNA is not going to be pulled incorrectly to one cell or another. It is important to check that also because you want to make sure that the spindles are attached. Because if they're not attached, then the DNA or chromosomes getting pulled in the wrong side. And then lastly, we want to make sure that they align perfectly at the equator, which again implies the health of the actual spindles. So to again highlight the importance of or understanding that the cell doesn't just randomly go through cell division, creating two cells from one cell. You do have a lot of checkpoints during that cell cycle. Now let's talk about telomeres, guys. So telomeres are basically these structure, these repeats of DNA that have the following sequence. And these repeats are basically, again, at the end of the chromosomes, there are only 6 bases. Here's again the sequence. So T, T, A, G, G, G. And they can be at, again, their length are different, but depending on the cell, every time that cell undergoing cell division, so one complete cycle of interphase and mitotic fails, you lose about 50 to 200 of these telomeres at the end of your chromosome. So in another sense, every time cell division happens for that cell does get, the chromosome gets shorter by about 50 to 200 nucleotide bases. Now, after 50 divisions, and again, this is the not the average number per se. The shortened telomeres basically say, okay, we had done enough. This cell cannot be used during the process of cell division, and we need to basically stop dividing. So that marks the end of it. Because again, as these telomeres get shorter and shorter, that implies that the cell is getting closer to its end of its ability to undergo cell division. Now, sperm and eggs and bone marrow cells, as well as cancerous cells, the cells that are basically undergoing significant cell division, they do have what we call telomerases that prevent the shortening of these telomeres. So if a normal cell is continuously losing those telomeres over time after each divisions, these cells do not cut the telomeres, which means that you can produce these cells over a long period of time with many, many divisions throughout their lifetime. Now, for us, the sperm, egg, and bone marrow telomerases are extremely good for us because that means we keep our ability to reproduce and create new stem cells. Horrible for the cancer cells because that means cells that have not successfully completed their checkpoints continue to divide because of their elongated telomeres, their undamaged telomeres, and as a result, we can have the growth of cancerous tumors. Here is a depiction of what the telomeres look like. If you have them stained, those are the little yellow spots. Another thing that I do want to mention in terms of cell cycle, it's something to note that the rate of the cell cycle and its ability of the cells in a specific part of the body to continue replicating depend on what we consider hormones and growth factors. So if you look at the name hormones, kind of again think about your endocrine system, the system that is responsible for producing hormones. When these hormones are basically produced by the corresponding gland and gets released into our bloodstream, directly into our bloodstream, they can have a very specific effect on a very specific part of the body. Growth factors have the same capacity, however, they act more locally versus throughout the body like hormones do. For instance, a growth hormone, a growth factor considered epidermal growth factor or EGF is extremely important for your ability to develop new skin underneath the scalp as a result of repairing, for the purpose of repairing, sorry. We also do understand that there are 2 types of protein called cyclins and kinases. These interact inside the cells, so they're not external factors that get released into the bloodstream, but these are the things present inside the cells, and they activate the genes that the products will be important for the mitosis to happen. So basically, if you want to kind of think about it, you have cyclins that are produced by the cell, inside the cell, right? That activate genes that basically important for mitosis. And that means that you increase the rate of mitosis. Because if those genes are making your proteins and they are activated, they encourage the process of mitosis to also happen. The last thing we're going to finish this lecture with is the concept of apoptosis. So apoptosis is basically the concept of the cell being destroyed for either abnormalities or the fact that their telomere is too short, or other possible reason as a result of damage. The cell undergoes the process called apoptosis. So for apoptosis to happen, one of the most important things that you need to have is a group of enzymes called caspases. And caspases, they basically get activated inside the cell. They're produced by the cell and they get activated inside the cell. What they do is they basically start to destroy the enzymes that are important to maintain your DNA or damage your DNA or replicate your DNA. So you're knocking out the DNA out of the equation as the process of apoptosis starts to take place. You also start to destroy your cytoskeleton. Again, if you want to kind of think about it just in terms of cell cycle, it's the fact that the spindles or the structures that come from the cytoskeletons, microtubules, and allow the DNA to be pulled apart in opposite direction between the two cells. They're also responsible for movement of organelles. Sorry about that. Movement of organelles within the cells, keeping the shape of the cells. So basically maintaining what a cell looks like and how to move things within the cell. So you take that ability also off. You also take away the ability of a cell to connect to neighboring cells, which is also a really bad thing to take away because you lose the ability to create secondary messenger systems, to create things like desmosomes where you stay connected to your neighboring cells, and basically function in a group of cells within your tissue. And the most important one, which I think is absolutely fascinating, is they start attracting phagocytes. If you don't know what phagocytes are or don't recall, they're basically white blood cells that eat unwanted material. And in this case, our unwanted material are cells that are basically old or damaged that needs to be removed and removed from the circulation and the body. So these phagocytes basically, again, come in and start to take bites of the cell parts or fragments and destroy to little tiny fragments that don't really do anything for the cell. Now, as this process is undergoing, as the phagocytosis and the white blood cells that are eating the cell and pulling it apart, you actually form these little bulges that are called blebs. And blebs are very important because they are actually surrounded by a plasma membrane. So they are encased, basically, even the cells being destroyed. You're not releasing and dumping the material inside the cell to the external environment. You're still protecting them inside these little tiny bubbles that are called blebs. And this is important because it prevents inflammation of the tissue. If this process doesn't take place properly, if the apoptosis doesn't happen properly, then you are introducing the inside material of the cell to the external environment that can trigger inflammation. Here's a depiction of what that cell death looks like. Here's a cell that's basically starting to destroy. You can see that the cell starts to lose its shape. So it was a beautiful shape right here on the first image. And then as time passes by, cytoskeletons being destroyed, capsids are destroying the protein inside the cell. Then you start to lose the shape. And then eventually you start to form these, again, little fragments of the cells called blebs, and blebs are going to be eaten by the phagocytic cells, and those phagocytic cells basically eat whatever that's been given to them and break them down to the subunits that make them, which is basically harmful, not harmful for the cell. Death of the cell, again, as we talk about this, one of the things that comes into mind is what is it that makes a cell be marked for cell death, which is something that I already described, and what happens when you do have cancer, and what is the characteristics of those cells. So as we're talking about this, one of the things that I do want to mention is the concept of necrosis. Necrosis is basically a form of cell death. It's typically associated if you have an inflammatory response in that area. So the damage, the inflammation and the damage is a result or associated with the tissue itself, and that's what causes the cell to die. In case of as I mentioned earlier, mitosis and apoptosis have to work together. So if you have cells that need to be destroyed or they're not good, they undergo the process of destruction or apoptosis and get removed. Here is what is very dangerous when we talk about the cancer cells. Cancer cells is when you have, again, too much mitosis and not enough apoptosis. If you take a look at the picture you have on this image, you notice that the normal cell is what we start with, and you do have some sort of mutation or change in this cell, so you can see it in the coloring differences. And that abnormality basically, in many cases, knock off any change checkpoints that your cell would have. And instead of figuring out that there is a problem with me, let me destroy myself through apoptosis, it continued to replicate itself and make more and more of itself and create what we call a malignant tumor. On the top of this picture, you also see what a normal cell looks like, what a normal cell division through mitosis looks like, and how healthy the tissue would look like when that cell division takes place properly. So that marks the end of our lecture for mitosis. Please take a moment to look at your PowerPoint. Make sure you listen to the lecture that's related to meiosis coming up next. And one of the points that you always want to recall is that mitosis is associated with the somatic cells versus the gametes being produced through the process of meiosis. Please let me know if you guys do have any questions or concern about the lecture, and I'll be happy to provide you with an answer. Thank you.